grateful to be here presenting a discussion of the principles of metrology in biology for the Center of Excellence in Regulatory Science and Innovation, homed at UCSF and Stanford University. My name is Marc Salit. I'm the leader of a group at the National Institute of Standards and Technology, which is headquartered in Gaithersburg, Maryland. And that group is the Genome Scale Measurements Group, and that is part of the Material Measurement Laboratory of the NIST. The Material Measurement Laboratory gets to do the infrastructure to support measurement science and measurement results in material science, chemistry, and biology. NIST is a part of the U.S. Department of Commerce, and as such, my job is to make sure that the tools are in place so that genomics can get adopted for clinical and commercial practice in the U.S. economy. It's a tall order, and one of the things we're trying to do is take successes from other domains and areas into this field, which is really emergent over the past 15 or 20 years. It's now 2016. The nominal finish line of the Genome Project was about 10 or 12 years ago, and we're still working to finish that in an authoritative way and make it so that genome sequencing is a routine and well-behaved measurement. But the tools that we'll be talking about today, these concepts, these principles of metrology and biology, the concepts and resources, really are work that, based on work that developed in the 20th century in physics in a rigorous way, and in chemistry at the end of the 20th century in a rigorous way. I have several roles, and all of my cards are here on the table. I am a consulting faculty at Stanford in the bioengineering department and in the pathology department, and I am a faculty of that Center of Excellence in Regulatory Science and Innovation. And so I'm pleased to be putting this metrology principles lecture in the kit for work in this space. So I've developed over the years a series of modules, and we'll be focusing today on the concepts and the resources. The concepts really give you a background in metrology and chemistry and physics, as well as some methodology of how that is used and how it plays out. The resources are a series of guidance documents from the international standards bodies and the global network that makes up the keepers of the International System of Units, or popularly known as the SI, and that would be a joint committee on the development of guides in metrology, JCGM. Along with this, I will do some teasing of what's to come in real-life measurement exercise and a sort of deconstruction of measurement results that comes from a fun exercise that can be done in a classroom, and a discussion of the tools of practice that broke. Did that break? No. Great. Discussion of some of the tools of practice also to come, and trying to figure out how to take these tools and apply them to living systems and create a metrology of biology. So what is metrology? Metrology is all the stuff that goes around the measurement result, so that you can actually take that measurement result and craft it into data that can support informed decision making. So I'm going to expose my worldview, and that is, in a good world, decisions are informed with data, and those data can be rigorously evaluated for their veracity and applicability to the problem that the decision is being applied to. Data are the results of measurements. So my worldview, again, is how do we put the stuff around a measurement result so that you can turn it into decisions? One of the things we've done is is established. And when I say we, it's I'm glad to have been a part of it and to be a part of it, but it's, again, a global network of scientists and engineers and statisticians and leaders that have established principles and methodology that lets us do a sort of formal calculus of confidence. So what does that mean? There's a formal system. There's a set of systematic practices you can bring to bear on your scientific endeavor that will let you know how well you can trust your data. And I'm going to talk about the principles and concepts behind that today and pepper that with some examples. I'm going to start with an example that says, well, why do we care? And is this really just dry? And of course we know it's not dry, and we know we care. And we're at a point now where there's some really high impact social issues that rely on evaluation of quantitative data and results of measurements over long periods of time in order to establish public policy and help us make decisions of what we do and its impact on others in the present and the future. I'm going to zoom in to a pretty significant example here where we can actually, walk through this question of, has this child been exposed to lead and does this child need treatment for lead exposure? So the posit here is that this kid who looks pretty happy and playing in the mud, I was one of those, may in fact be playing around a house that's had, built in the 1930s and had 60 or 70 years of coats of paint on the outside of that house. That was lead paint, flaking off, turning into dust through weathering and through the process of people walking around at summer barbecues and crunching the little chips into dust. And anyway, the house has been remodeled recently, and so there's also a possibility that there's tons of dust around from that remodeling. So there's this question that came up, and the child's parents and the physicians who will be drafted for the clinical intervention, if there will be one, have to decide, was the child in fact exposed? Is treatment necessary? And if it's needed, what is the appropriate treatment? How does this calculus of confidence play out there? I want to be confident in the measurement results. I want to be able to know that the results can be meaningfully compared through traceability, metrological traceability. I want to know, gee, if I measured this again, what's the likelihood that I'll get a result that is equivalent? So that there's a way of doing that we traditionally call estimation of measurement uncertainty. And I want to make sure that the lead measurements are the right lead measurements. Would I use a different tool to measure lead at the sort of 10 ppb, 10 micrograms per kilogram in this child's blood, which is the action level? Is that the same measurement tool and measurement process that I would apply to measuring lead in some lead ore or in some alloy that has 30% lead? So what can you do to give me evidence, in fact, that the measurement I've made is sensible and that I should move forward with decision making based upon these kind of results? And ultimately, have there been quality systems developed and applied all throughout this enterprise? Because what you're just talking about here is a whole enterprise if you want to figure out how to treat that child. We need a way to understand, and we posit that there are systematic approaches that can help us do that, and live in a world where, in fact, decisions are informed by measurement results that have an established confidence. So this is this sort of four-legged stool I show here. I prefer personally a three-legged stool, and the audience that has been paying attention will see which leg I pulled off. And that leg, I'll go back and forth, and you can see that what I pulled off was quality systems to get to here. And I'll talk a little bit more about why that is. Quality systems have been really promulgated post-World War II, but in the measurement science domain, quality systems have really been put into place in the mid to late 1990s with the adoption of ISO document 17025, which called for all testing labs to make statements around traceability of their measurement results, or metrological traceability, and to do quantitative estimation of measurement uncertainty. It's all of a piece, and one thing I will say is that the quality systems element of this calculus of confidence remains emergent, and in a regulatory science domain is an essential piece. So I show it here. And I think, though, it's a non-technical but rather process-oriented enterprise approach, and it is not like the other things, which really are technically based. So I will continue to discuss it. I won't beat a dead horse with that fourth leg of the stool, but I do prefer a more elegant formulation of the physical science-based calculus of confidence. And as I'd already noted, the three legs that I'm focusing on are metrological traceability, estimation, quantitative estimation of measurement uncertainty, and method validation, shortened there to those three single word descriptions. I'm showing here the exact definitions from a guidance document called the VIM. The VIM is the International Vocabulary of Metrology, and I won't pronounce the French version. This is actually a diplomatic document where there is a French language version of this dictionary in parallel, side by side with an English language version, and where there is a conflict in the terminology or in the definition that arises between those translations. The French version is the authoritative version, and that's in part why it's called the VIM, because it's the French acronym for International Vocabulary of Metrology. This comes from the group, the JCGM, which is the Joint Committee on Guides in Metrology. So I'm going to actually go through these definitions, and I'm going to give you my just sort of worldview on those as well. Measurement uncertainty is a non-negative parameter characterizing the dispersion of the quantity values being attributed to a measurand based on the information used. There's extra words in there, I think, but the important thing to understand from that definition is we're trying to characterize the dispersion of results that are likely attributable to a measurement result. So that measurement result is a quantity value, and it has some distribution. The story about measurement uncertainty is that it is a way that I can quantitatively understand the likely dispersion of a measurement result about its value or its reported value. So if I tell you a measurement result is 33, I'm not telling you everything I ought to know if I want to, again, make decisions. Or let's go back to that example of the child who we're asking, does this child need treatment for lead poisoning? The threshold for no treat is 10 ppb, or parts per billion. If I make a measurement and I measure 11, does that mean that kid's in the clear? And if it's 11 plus or minus 4, then I have to really think more carefully, because what I've just told you, the plus or minus, is the likely dispersion about 11. So we're going down to 7. So I'm telling you that it's somewhere between 7 and 15. because I've got an uncertainty bound. And much more nuanced than just that number is that limit from 7 to 15 that I've told you about my measurement, I have to tell you something about the likelihood that it's 11 and the likelihood that it's 7. Is that what we call a 95% confidence interval of a symmetric distribution, or is it much more likely to be between 12 and 15 than it is between 7 and 9? So there's a lot of information, and it's a vague enough definition here that one can actually build the metrology to make sure that you're delivering a result that is appropriate for its intended use or fit for purpose. So it's a non-negative parameter, and in fact it is describing a distribution that is attributable, or a dispersion. The other thing to note here, and it's kind of geeky, but it needn't be a single number. It could in fact be a range that describes the distribution, and it needn't even be symmetric about the result you're reporting. Like you might be able to say it's much more likely between 12 and 15 than between 9 and 11. So there's a lot of ways that measurement uncertainty can usefully be used. It's important, as a leg of the stool, it's important to be able to properly, for the intended use, describe that likely dispersion. Metrological traceability is, conceptually, it's how did I get the units on this thing? How do I know I'm reporting out parts per billion lead? I've put this blood sample in a instrument that gives me a readout. Whatever the principles of that measurement were, I need to know that if I measure that lead in blood on that instrument in this laboratory on this day, that I can compare it usefully to other measurement results. In fact, most importantly, the measurement results that the panel who decided at the action level for treatment of lead poisoning were looking at a comparable scale of measurement results. So that's how we get our units on things, is through metrological traceability. We usually use a method called calibration to do that, to convert from instrument reported units into a common shared unit that's traceable through an unbroken chain of comparisons to a shared reference. And so the exact definition in VIM currently, as of 2008, which is the VIM 3.0 guide, it's a property of a measurement result whereby the result can be related to a reference through a documented, unbroken chain of calibrations, each contributing to the measurement uncertainty. And critically, of course, if I calibrate, that calibration process creates its own dispersion about a measurement result, and one has to propagate that dispersion back up to the measurement uncertainty around the reported result. There's an older definition of metrological traceability that has a few less words that I'm going to show in a moment, which I prefer, and it's the 1993 version of the VIM, and I think it's a little cleaner. Validation, and then we'll go on to my cranky response to the newest version of that definition. Validation is the provision of objective evidence that a given item fulfills its specified requirements where the specified requirements are adequate for an intended use. And that's a lot of words to say that what I'm trying to do is build evidence that you should trust this measurement result. And it's the process, and it's a formal process, typically subject to a critical review that lets me establish the veracity of my result. It says, not only is this method good for measuring lead in blood, but I can show you evidence that if I had a 7 part per billion lead in blood sample and an 11 part per billion lead in blood sample, I could tell the difference between those and compare them against the 10 part per billion action limit and meaningfully make decisions. So it's that it's that process whereby I build a case for you to trust me. It's age-old human practice. The old-timey definition of metrological traceability is that it's the property of a result, the result of a measurement, or the value of a standard, and that's kind of a dorky, again, I think it's overly complex, whereby it can be related to stated references, usually national or international standards, through an unbroken chain of comparisons, all having stated uncertainties. So it's really important here to highlight the essence of traceability, metrological traceability. It's the property of a result, so a result can be traceable or not. And the result of a measurement, or the value of a standard, so the traceability could be of a particular artifact or object to a particular other reference. So you've got a reference here, and it says can be related. Your result, the result or the value of a standard can be related to a stated reference. And parenthetically, it's usually a national or international standard. Well, I want you to understand very carefully that the people who developed this definition have a conflict of interest in pointing you to national and international standards because that's their business model. The fact that it says usually a national or international standard, that's just advertising. The cool thing is that I can share a artifact with my friend in the lab down the hall, and the two of us can actually make results that have metrological traceability to that artifact we just shared. And it could be a calibration solution that one of my graduate students made, and we split it in two and gave a sample to each lab. So we can compare results now between our two labs. We don't need to know much more about that calibration sample except that we're using the same thing. So that's now our stated reference. And then we can, in fact, do science together. So I'm traceable to that calibration student that graduate student made, or my measurement results are traceable. And that's where we move from. How do I make myself, or how do I make my measurement results? Sorry for falling into the same trap I accuse people of. By the way, precision in language is one of the tools we use to be able to have this international measurement system. And that, frankly, going back up the tree of my slides to the JCGM guide here, we get so serious about precision of language that we share a document amongst ourselves that says, when I'm talking about metrological traceability, this is what I'm talking about. So I can, in fact, say that I'm talking about a property of a measurement result. And when I get sloppy and say I'm traceable, that's a meaningless concept in metrology. But I can say that the measurement results that I am providing from my laboratory, from this experiment, are traceable to this particular artifact or standard. So in order to get traceability, metrological traceability on my measurement results, I need a standard and a way to compare to that standard. So we can go back in time to the units that were defined somewhat egotistically by emperors and kings, where they might define a yard as the distance between the tip of my nose and the tip of my finger. And when that king is no longer king, we'll have a new yard. The yards are about comparable to each other because they're human size. And that's how we got, in fact, to a agreed upon set of references. But it can break things in a measurement enterprise when the references shift under the ground. So I promised to get back to talking about quality management systems as this fourth leg. That maybe I should start calling it the shadow leg of the stool. It's a different kettle of fish. You might have heard about ISO 9000, and it was this series of documents that really brought quality systems and quality system development into common practice and common awareness across manufacturing enterprises. And what has happened in the measurement science world is that those same principles have been adopted into laboratory practices. And again, it's probably worthy of a CERC lecture or so on how quality systems form the basis for FDA regulated good manufacturing practice and good laboratory practice. That's the kind of stuff I'm talking about here, and it refers to what an organization actually does to manage its processes, what it does to manage its activities so that products or services from that organization meet the quality objectives it has set for itself. And one of the important things throughout quality management systems is that it's a documented process. So you say what you're going to do, and then you do it, and quite often there'll be a log or transcript, in fact, that for a given measurement process, in this case, what we're talking about today, that process was followed through. There are all sorts of benefits that arise from quality systems that have really come to the fore in enabling better results to be shared and better decisions to be made. I have here a cartoon that shows it as a different kettle of fish, but really one of the things it has done for us, and I'm going to skip ahead a couple slides, and this is a Dilbert cartoon. My colleague Dave Dewar is a lover of Dilbert cartoons, and he made this slide with me. It lets us catch our blunders. One of the things that metrology doesn't have in the three-legged stool is a way to deal with blunders. Oops, I forgot to add this reagent in this process. Oops, I didn't check this, and we ran this at the wrong temperature. Systematic processes work really well for this, and having checklists and having documented processes, standard operating procedures and technical procedures. Another critical aspect of what we get is institutional learning and memory. And that the longer I go at the science I do, the more critical I think this is as an element. If we can develop measurement processes and results that are independent of the individuals carrying them out, that's the kind of system I want behind the decision making going into clinical treatment and clinical outcome for my child. So there's an emerging consensus. The US adoption lags that adoption and practice in Europe and Asia. And one of the things that is powerful, though, and is a force that you should be awake to, and especially working in regulated context, is laboratory accreditation as a way to establish the existence of a quality system and compliance with that quality system. Laboratory accreditation does a lot of things, and that's one of them. I'm going to go backwards in time now and talk a little bit more about metrological traceability. I didn't really go backwards in time. That was, so I talked a little bit about getting units on your result. And here is a tease on the formulation of using the concept of metrological traceability to get comparability. Here's a simple model. I've got a chemical analysis equation here where you're measuring the concentration of an unknown and comparing that to the standard for that unknown. I'm comparing it to the standard for the unknown here, and you've measured the signal for the standard, you know the concentration for the standard, and you have the signal for your unknown. You can actually do a fix to your calibration if you've got a model here that allows for a recovery factor. And now I've got a measurement model that's a quantitative model, and I can now establish metrological traceability to that standard. and results that are compared to a standard that standard is interoperable with can be compared to each other. So what we've done is calibrated there. One of the things I haven't talked about yet is a really basic concept, and it's one of the ones I want you to walk away with today, is that measurement results are only useful when they're compared. They're not even interesting when they're not compared. I'm comparing two measurement results to each other to see, are these two conditions different? If I'm testing a hypothesis that says they should be different, now I have a way to evaluate that with a measurement system. Are they in fact supposed to be the same? In other words, my hypothesis is this treatment should have no effect on that measured parameter. I need to be able to compare the measured parameter then with and without treatment, before and after treatment. In other words, I want to observe a trend, or this should increase over time. I've got a hypothesis that says that in my science, or in fact, I'm treating a patient with a therapeutic, and I want to observe the trend of the response to that therapeutic. I want to be able to compare measurement results today, and I want to compare it to those measurement results tomorrow. I need to know that those measurement results, in fact, are both traceable to a common reference so that they can be meaningfully compared. And I show here a cartoon of the ultimate comparator tool, the two-pan balance, which goes back to Greek times, I believe, or predates that, where in fact people were trying to barter one substance for another or one object for another. I want some eggs and you want some chicken. Let's trade. And so this lets me do comparison right in the moment. And what we're doing here with metrological traceability is, in fact, having a shared object that lets me compare, in this case, the mass of the person against the mass of the gold. I want to then compare the mass of a sheep against the mass of the person, and I'll do that through a shared mass of gold. Different places, different times, measuring different objects. The idea here is that through metrological traceability, we get comparability over space and time. And by comparability, I don't mean these two things are about the same, but I have the ability to quantitatively compare this thing against that thing, where this thing is a measurement result and that thing is a measurement result. So comparability of results is the whole and sole goal of metrological traceability. There have been allusions around metrological traceability doing more than that. In fact, there's an often mistaken idea that metrological traceability can get you the right answer or get you accuracy. But it doesn't do that. It just gets you comparability. So results linked to a common reference can be compared, and the scope of that reference, very importantly, can define the scope of comparability. So I talked a little while ago about comparing results from my lab and my colleague's lab down the hall by sharing a common calibration solution that was made up by one of our graduate students. I don't need to know much about that as long as all I want to do is compare results from my lab and the lab down the hall. But if I want to compare results from my lab to a lab in a different location, a lab in Washington, D.C., I need to, in fact, either share that calibration solution with the folks there or make my calibration solution comparable or traceable, have measurement results that are comparable to a shared reference somewhere else. And often we use this global network, which is the International System of Units, which gives us the basis for a harmonized, stable, maintained, I would even go so far as to say well-maintained by a network of national measurement institutes from around the planet, set of units. And we quite often in chemistry and biology rely on the SI for units around time, temperature, mass, abundance, or mole. and electrical units when we're measuring electrical properties in a biological context. I'm going to talk next about measurement uncertainty and practical realization of measurement uncertainty. So often I'm faced with the problem of comparing results again here. I want to know if those two purple dots are the same. And I can't tell you whether or not they are. With that information that's on that graph in the right-hand part of that slide, we don't have sufficient information to inform a well-based decision. I need something like this, where in fact what I've just done is in this graph using a common vocabulary for describing measurement uncertainty, the error bar around a dot on a chart. I've described the measurement uncertainty or the reasonable dispersion I can expect around those two measurement results. On the top, I can't tell you anything about whether or not these results are the same. On the bottom, I can tell you that very much they are the same, assuming that the statistics used to generate those error bars are meaningful for the comparison I'm making. And so there's turtles all the way down on the nuance of a measurement uncertainty estimate. And a lot of that can be simplified as long as I say, are these results good enough? And so it's a completely arbitrary graph, and that's done purposefully. There's no axis on the y-axis. All I can say is using the common vocabulary that we have for measurement results and representation on a graph, those two are the same, fit for purpose. I want to zoom in on measurement results here and plant the seed for further discussions in further modules, further lectures. The idea is that all of my measured data are samples from a distribution. This is especially important in biology, where often the distribution of my measurement results are underrepresented if I've only sampled a few biological conditions. There's an element of measurement science in biology where we often don't get to really see or unpack the dispersion arising from measurement and the dispersion arising from what is commonly attributed to biological variability. So what's important for us to note today, the thing we're measuring has some underlying distribution. We sample that distribution. If I'm measuring lead in this child's blood, there may in fact be a different amount of lead in blood at different times of day. There may be a different amount of lead in blood over a period of time, or if I've got triplets that were out in the yard playing in the dust, or 3 different kids, they may each have a different amount of lead in their blood based upon how much they like to eat the dirt or who was at the bottom of the pile when they were rolling around. So there is an underlying distribution. It's important for me to remember that the things I'm measuring are not typically completely homogeneous and pure. And our sampling of those things is typically fraught with extra dispersion and biases. So a lot of what we're trying to do is separate out the sources of variability and estimate their relative magnitudes and try to understand when asking the question, and this is where the nuance of the measurement science goes, when asking the question, are these conditions the same, or can I make this decision based upon the data that underlie it? Understanding this distribution or these underlying distributions is key. And really what you're doing, and this is sort of a Bayesian model, is you've got multiple things that go into my measurement, and they each have their own distribution. Here these are shown as probability distribution functions, or the likelihood of a value. The values are on the sort of cartoon x-axis for these PDFs, and the likelihood is on the y. So here you've got what's typically familiar to us as a normal or Gaussian distribution on the top. This is a trapezoidal distribution, sort of a top hat distribution. Something like this might be, in fact, the likelihood you're delivering the right amount in a pipette. This might be the tolerance that's given to your pipette, and there's some likelihood that you're out of tolerance in any given pipetting action, but you're most likely you're just as likely to be 5% below as 5% above, but you're unlikely to be 8% below or 8% above. That's sort of a nominal distribution of a trapezoid there. And here's another kind of commonly assumed shape where, in fact, I have no information about the shape really. I know that the most likely I'm in the middle with a triangular distribution, and then it becomes very unlikely out beyond a certain range, and I'm going to say that it's a diminishing likelihood in a linear way. We don't usually have good information when we are making that assumption, so I will say that this is cartoony more than informed with data. Needless to say, if you've got all these underlying distributions in different elements of your measurement process, they combine in some magical mixer way to give you the distribution of what you're trying to measure, or the measurand, as it's noted here. This magical mixer is the measurement model or the equation that takes the input data and gives you a result. I am going to touch on how we got here with a couple of slides from some earlier work in establishing measurement science and metrology principles in chemistry. And just want to give you a teaser that doing this work in biology is emergent. In chemistry, often you want to say, I've got lead in blood here. How do I know I'm measuring lead and not some other metal? So the identity of the thing you're measuring becomes much more challenging than in measuring physical properties. Physical properties might be mass, it might be length. I know when I'm measuring length, circumference gets a little more difficult than length, for instance. It's much more hairy when the substance you're purporting to measure, in fact, has to be identified as being the right substance. I need to prove to myself and to the consumers for my data that I've not got an interference to my measurement result, and in fact that I'm measuring something that represents what I'm interested in physically. If I'm telling you this is the lead in blood, what does that say about the lead in the cerebral spinal fluid, which might in fact be the important thing to understand. So traceability in chemistry is different, and we developed systematic approaches to do method validation to establish the scope of validity of those measurements and to present a clear description of a measurement model that gives you that objective evidence that what I've told you I'm measuring is in fact what I'm reporting, and that the result is robust with respect to the interferences that I can expect. So we check that measurement model with method validation to test that it's complete, to test and evaluate assumptions, and to help us establish, in fact, the understanding I get from a validation experiment or series of validation experiments helps me to establish the uncertainty budget. It also identifies what parameters need control in order that my calibrations are valid and I can assert metrological traceability, and that the work I've done to estimate measurement uncertainty applies to the measurement result, in fact, that I'm reporting, not just in general. When I use this method, it's good to 20%. And this method validation would also systematically test the scope of that validity. All right. So I am going to skip the next two slides and get to why you should care. Again, we've got decisions to make, and they're critical to make. So we've discussed the primary principles. I've touched on a few elements of the infrastructure, and we'll make sure that materials are available to describe that more completely. There are some essential tools that other modules in our series will present and emerging challenges, which is the stuff we don't yet know how to do systematically. I want to mention that at this point we consider metrology to be something of a craft. Two metrologists, quite importantly, two metrologists might in fact use different strategies for calibration to achieve metrological traceability. They might do different experiments to evaluate measurement uncertainty and estimate that. They may do different experiments and have different practices to establish method validation. The idea at the end is that the results from two different metrologists would be comparable and equivalent when the rubber meets the road, when the decision making happens. You can mix and match data from either of those processes. So what can you walk away with from what we've covered today, and what can I tease you with that will have you back for the next lecture? I want you to walk away understanding that you can't trust any measurement results without having all the stuff around them that metrology will give you that lets you make good decisions with those measurement results. Ultimately, you need to compare measurement results to use them in a scientific inquiry or a decision-making process. So we're trying to provide you with the approaches for critical evaluation of those measurement results, how to use the existing global metrology infrastructure, and we'll have more on that with more discussion of resources that are available and how those can be applied, as well as what's emerging that will enable you to practically apply the metrology principles in your science, in your regulatory science, in your regulated applications, and in building frameworks for decision making. Ultimately, we will invite you in to see how we're working to extend the global metrology infrastructure to support decision making using data that is from the biological context. I really want to close with giving you some ideas of what we don't know how to do yet. We don't have all the answers at present. The metrology of quantitative measurement science in biology is in its infancy. We're using a lot of tools that give us the best answers we can get at the time, but may not be definitive. We use collaborative studies, in other words, multiple people making a measurement and seeing how those compare, multiple people, multiple labs, multiple approaches to measure the same phenomenon and see if we can, in fact, get harmony of our results. We use reference systems, in other words, laboratories that all promise to do the same thing. Even if we're not evaluating them, we try and hold them all to the same protocol. Sometimes that's what we're using to get metrological confidence in measurement results. We've got international standards and a growing effort amongst the National Measurement Institute network. I would invite you, I think NIST is at the forefront. I would invite you to look at the resources and products available from NIST for your measurement science needs. And the concepts, and this has been my hypothesis for 15 years or so now, the concepts from traditional metrology absolutely apply in the biological sciences, and we're trying to demonstrate that and build the tools and extend the frameworks to make that happen. And we need still to develop some good practices that remain to be hammered out to apply those concepts in biology. I want to acknowledge the infrastructure at NIST that has supported development of this practice of metrology in biology. I want to acknowledge, most importantly, a colleague, David Dewar, who's in the Chemical Measurement Science Division of the Material Measurement Lab. He and I have been working for 25 years together on formulations, first in chemistry and now in bioscience. And a lot of the material we've presented today and will present in future lectures is based on some coursework that Dave and I have prepared and presented in a number of venues. So I look forward to next time we get to meet and moving forward on discussion of resources and opportunities to apply some of the tools we've got to some real data. Thanks.